Today I'm going to talk to you about epigenetics. Epigenetics means literally above genetics, and it's the study of everything except our DNA itself that allows our genes to get expressed. It's a relatively new field. It really only started in the 70s, but now there's a lot more focus on it because it's giving us answers to things that we previously haven't known about. So epigenetics determines gene expression in cells, and it has nothing to do with actually changing DNA, which is a common misconception. It's really just about the chemistry of the molecules on the outside of DNA. And epigenetics is really good at explaining nurture in the nurture versus nature argument about why we are the way that we are, because our nature is our DNA, which is unchanged. The base pairs themselves and their order is unchanged, but nurture is what we do, and that impacts the way they get expressed. One way that epigenetics changes the way you express your genes is through a process called methylation. And methylation patterns can be changed by many things, like by smoking, toxin exposure, other natural processes like pregnancy, going through puberty, diet, exercise. They also contribute to the way our genes get methylated. And one example is an experiment done with 23 men who did not exercise at all, and their methylation patterns of their genes were mapped before and after 6 months of trying out exercising. So what they noticed was that the methylation patterns changed in the genes related to obesity and type 2 diabetes. There were about 1,315 sites in the type 2 diabetes genes and 24 sites in the obesity genes that changed patterns to have more methylation, which allowed the suppression of factors that could cause obesity and type 2 diabetes. And how these different methylation patterns can change the way genes are expressed, I will get into in a little bit. Methyl groups tell DNA how to act by regulating their transcription. In essence, they're an on and off switch. The methyl groups are specifically attached to cytosine molecules, which are located at. Methyl groups tell cells how to act by regulating transcription. In essence, they're an on and off switch. Methyl groups bind to cytosine molecules, and they occur specifically at CpG sites, which is where cytosine is next to a guanine. And this is important because both of the cytosine molecules need that methyl group so that as DNA is replicated, the methyl groups can be replicated along with it. So, in a strange way, epigenetics, in addition to just DNA, is also heritable. Methyl groups regulate transcription by changing the way that enzymes are able to interact with the DNA because they change the structure of the DNA. And a common theme in biology and chemistry is that structure determines function. So the change in structure by adding or subtracting methyl groups will change the way that enzymes function with it. If you view this diagram, you'll see that there are two main ways that methylation can control transcription. First, these CpG sites where methylation occurs, they're usually before promoters on genes, and promoters act as signals to tell enzymes where to start transcribing so that proteins can be made. And what these methyl groups do is they kind of stick out in funky ways and tell enzymes basically to go away, and that ends up silencing the gene because there's nothing to transcribe it, since the enzyme doesn't really fit anymore due to these new methyl groups. Another way that methylation stops transcription is by recruiting other proteins like histone deacetylases to aid it in stopping other enzymes from binding and translating the genes so that they become expressed. But I'll get into histone deacetylases a little bit more later. So you might be wondering, how exactly does this methylation occur? And the main way is through enzymes. So DNA methyltransferase is an enzyme that's responsible for methylating the cytosine. What it does is it flips the cytosine 180 degrees out of the DNA so that rather than interacting with the guanine, it is out in the open, ready for reactions to occur. What happens is the DNA methyltransferase obtains a methyl group from a molecule called SAM, and it attaches it to cytosine, turning it into 5-methylcytosine. And when demethylation occurs, human TET or 1011 transferase is able to do this through a series of reactions that starts with adding a hydroxyl group to it. And other reactions, there are a couple of them, but after doing that, it's able to return the molecule back to regular cytosine. The second example I want to share about methylation is cancer. So DNA methylation, it's usually regulated by the DNMT. There's a few kinds. There's DNMT1, 3A, and 3B. The main difference between them, which I won't get into too much, is when they occur. So 3A and 3B are for early cell differentiation when you're a zygote, and DNMT1 is for cell division past that point. In cancer cells, though, things are different because DNA methylation patterns get disrupted. This happens in two ways. So there's hypermethylation, which causes the silencing of genes, and an example of this is tumor-suppressing genes. So when those suppressing tumor genes are inactivated through methylation, that tells the body to make tumors. So that's why when you have cancer, there are large masses of cells that are aggregating. It's because that gene gets turned off. Also, widespread hypomethylation causes new genes to get activated or turned on. And some of these new genes are responsible for it having increased capabilities to reproduce or degrading cellular matrices. It allows them to move more efficiently, so metastasis can occur and the cancer can spread from one place in the body to another. There are a lot of differences between cancer genes and cancer cells and our normal cells, and a big part of that is not only the genes. The difference is not in the DNA. The difference is in how the DNA is expressed through epigenetics. Besides methylation, histones also play a role in why cancer cells are able to behave differently than regular cells, and just in general why cells are able to be differentiated from each other. So histones are proteins that combine with DNA to make chromatin, which is what you usually think of when you think about our chromosomes in that thick shape. What histones do is they're able to condense them, and through histone deacetylation, we're able to condense the proteins more and the DNA so that less transcription can occur. And this happens through deacetylation, which is taking the acetyl functional group off of a lysine amino acid. And by doing this, we make it so that the proteins are wrapped more tightly together, which in turn limits the accessibility of regions of DNA for transcribing enzymes to read. So, in essence, more histone deacetylases corresponds to less transcription, which you can see in the image below, because there's simply not enough room for transcribing enzymes to come in and work with the DNA. Epigenetics was particularly interesting to me because we got to learn more about structure and function of molecules and how that interacts with our biology. And I thought it was pretty interesting because that was a theme common in Napoleon's buttons. For example, in the birth control chapter, I learned that there was really not that many differences between testosterone and estradiol, and that really only 3 changes, one of them being adding a methyl group, could make someone exhibit the characteristics of a different sex. So, yeah, I just think it's really interesting that just by adding methyl groups either to a molecule like estradiol or to DNA, it can have profound changes even though it may seem small. Something to note, though, is that steroids aren't polymers, so adding just one methyl group can make a significant difference for a steroid, but one methyl group really won't change a polymer too much, and that's why widespread methylation and demethylation needs to really occur if we want to see fairly drastic changes in the way that genes get expressed. The polymer example earlier was DNA, so we were modifying that polymer to exhibit different characteristics, but a cool example that I found was in polysaccharides. And polysaccharides are just units of sugar molecules linked together, which is more similar in structure to DNA than steroids are, just because there are repeating monomers that get linked together to form the polymer. When talking about DNA and epigenetics, we were adding methyl groups to the nucleic acid polymer, but here we're adding ether groups to the cellulose polymer, and it's actually quite viable for us for a couple reasons. First of all, adding this oxygen makes it have more energy stored, and this energy we can use to combust and get energy out of it ourselves. So this plant is actually very viable as a source of biofuel. Also, it's able to be used in commercially producing food and drinks because we're able to use it as an emulsifier now that it has these new properties given to it by modification of adding a couple functional groups. Hopefully what you got out of this is that while big molecules have a specific structure that determines their function, there are also minute details of it, like adding functional groups, that can really change the way that it's able to function and the way we utilize them. Thank you for listening, and I hope you thought it was interesting.